Holly, and my role is a food LCMS scientist with the global technical marketing team here at SciX. So I hope you've enjoyed and found the previous talks useful. For this last talk, I am going to give an overview, as Francis has already mentioned, of the different types of PFAS exposure pathways and what we here at SciX have done in terms of developing analytical methods to support the analysis needed in the matrices that are involved in these pathways. So, as we have already heard from several presenters today, and we've already seen this life cycle picture of PFAS, due to their widespread production and use of these chemicals, people can be exposed to PFAS in a variety of ways, such as through drinking water, food and food packaging, the use of household and personal care products, being close to waste sites like landfills, wastewater treatment plants, the use of firefighting foams at airports and military bases, and as well as the application of biosolids, commonly used as fertilizers for amending agricultural lands. And also, this practice also impacts the ground and surface water that are collected near these sites, and also the produce and the animals that are harvested from these lands as well too. So on October 18, 2021, the US EPA announced the agency's PFAS strategic roadmap, which really outlines the agency's approach of different actions and timelines that EPA is planning to enact and enforce new policies. Really, the goal is to safeguard the environment and communities from PFAS contamination. And the roadmap accounts for the full life cycle of PFAS and the different pathways of exposure. And it's evident by the number of different agencies that are involved, as I'm going to speak to it here. So, as we've already heard mentioned in Craig's talk earlier, last June, EPA announced both interim and final health advisory levels for four different PFAS: PFOS, PFOA, PFBS, and GenX in drinking water. And right now, the agency is working towards developing national primary drinking water regulations for these chemicals, and they're expecting to announce the proposed levels soon for public comment. Similarly, Health Canada has also proposed an objective value for monitoring PFAS in drinking water at a concentration of 30 ppts for the total amount of PFAS measured in drinking water. Other exposure pathways that are currently being worked on by the agency are wastewater discharges to and from municipal wastewater treatment plants and their impacts on surface waters. There's also a lot of ongoing data collection that's being done in fish and also in biosolids and air as inputs for risk assessment studies to understand the fate and transport of PFAS through these matrices as well too. So as I'm going to talk about in the next couple of slides, human exposure to PFAS is going to start taking more of a center stage with a lot of focus on exposure pathways that are a lot closer to home, like consumer products such as cosmetics and textiles, and food packaging, all of which are starting to get a lot more regulatory attention, such as the ban on PFAS in cosmetics and apparel in several US states, as well as the US FDA's increased engagement with organizations like the AOAC International to develop new standard methods for analyzing PFAS in food. So here's an overview of SciX's past and recent response to these global PFAS trends, as we're leveraging our portfolio of hardware and software products to develop methods for analyzing PFAS in different matrices. So as you can see here, we do have a wealth of technical materials on PFAS analysis in various water matrices. Most recently, leveraging the sensitivity from our newest nominal mass system, which is the SciX 7500 system, to reach the low detection limits needed to analyze for PFAS in drinking water, groundwater, surface water, and even seawater. But like the EPA, we have also taken the PFAS life cycle into consideration when it comes to developing new methods, especially as we start moving beyond water matrices into other more complex ones, such as firefighting foam, human blood, consumer products like cosmetics, textiles, food packaging, and food as well too. So given the new and upcoming state and federal regulations, most of which are directed at consumer products in the cosmetic, packaging, textiles, and food industries, for the next few slides, I'm going to discuss some recent work that we have done at SciX on these exposure matrices, most notably in AFFF foam, firefighting foam, several consumer products, food contact materials, and in food. So first, I'm going to talk about PFAS and firefighting applications. So, just as a bit of a background, and we've already heard the story from Craig during his history review of PFAS. As we all know, back in 2001, there was a spill of AFFF foam in Etobicoke Creek near Pearson Airport, which is located in Toronto, Ontario. And at the time, fluorine NMR and LCMSMS were the two analytical methods used to analyze this contamination. And at the time, only three PFAS compounds, the C6 sulfonate, PFAS, and PFOA, were the only three PFAS that were identified using targeted LC-MS analysis. So fast forward 10 years later, no surprises that AFFF foam we've discovered is a lot more complex than just these three compounds, where we have a number of different studies using high-res mass spec, and they found identified more than 40 classes of PFAS, including both legacy and novel PFAS, some of which are precursors that can degrade to PFCAs and PFSAs. And this knowledge gap becomes even more evident when we start using other techniques like total fluorine and total organofluorine assays to measure the total fluorochemical burden in environmental samples like surface water that has been contaminated with these AFFF foam. And what we typically see is that the target analysis of known PFAS usually only accounts for about half of what is being measured. And this really points to the fact we do need better non targeted screening tools to fully characterize this remaining PFAS dark matter that is currently escaping our targeted techniques. So in this work, I'm showing what we've done is we use the Xenotop 7600 system, specifically its new fragmentation mode called electron activated dissociation, or EAD. And this technique involves bombarding the plume of sample ions entering from the Q1 of the mass spec into the EAD cell with free electrons at a certain kinetic energy. And what this does is it promotes these precursor ions into a radical state followed by fragmentation into a suite of fragments that can then travel through the mass spec and make their way into the top analyzer for detection. So compared to the more conventional collision-induced dissociation, or CID, which typically cleaves compounds at their most labile sites, EAD is a much softer fragmentation approach, and this usually results in the creation of more diagnostic structural fragments. So in this example here, we use an EAD method to analyze for these two fluorinated surfactants in a firefighting foam sample. And the resulting MSMS spectrum was then compared to one acquired in CID mode. And if we overlay these spectra together, we can see that EAD here, shown in the blue up here in the top, it gives us many more unique fragments than CID down here in the pink for these two PFAS. And this really helps us increase our confidence in identifying these compounds during targeted analysis. But as well, if we're doing non-targeted screening, it helps provide a lot more structural clues when we're trying to figure out the structure of unknown compounds. So next, I'm going to give a brief overview in consumer goods. So over here on the left is a snapshot from a review paper by Julianne Gluge et al. And they looked into a massive amount of published literature and patents on where PFAS are used for what applications and which specific PFAS. And the authors identified more than 200 use categories for PFAS and attributed them to over 1,400 individual PFAS. And this has been one of the more comprehensive review of both essential and non-essential uses of PFAS. And of these 200 use categories, apparel and personal care products were listed, both of which have been reported to contain a variety of different PFAS in recent literature. So the top figure here shows the concentration distribution of PFAS observed in 38 cosmetic and personal care products from Keegan Harris, a former PhD student of Professor Amy Rand at Carleton University, which we will hear speak more on this topic tomorrow. The bottom figure here comes from a study published last year that analyzed over 70 pieces of children's textile clothing that have been marketed as stain resistant from both the States and Canada. And using a combined approach of particle-induced gamma ray spectroscopy for total fluorine and then also targeted LCMS and GCMS analysis, a suite of different PFAS were identified with higher concentrations typically observed in school uniforms and outdoor wear. And both of these studies were very timely considering the recent and upcoming regulations on different consumer products in various states. And we're also starting to see a lot more media coverage on findings like these. So here in SciEx, using the SciEx 7500 system, we developed a method to analyze various textiles, and we observed the presence of three different PFAS, including the 6,2-fluorotelomere sulfonate and the 6,2-dipap in some of these products. On the right here are results from using the Xenotop 7600 system to perform a non-targeted screening of various cosmetic products. And consistent with the findings from the Harris et al. Paper that I just mentioned in the previous slide, we also reported positive hits for the 6,2-dipap in almost all of the samples we analyzed, with low precursor mass error, isotope pattern match, and also great library scores as well too, using our SciEx fluorochemical library. And what's more is that we also see detects for the mixed chain length dipap, such as this one shown here, which is characterized by a phosphate head group attached to a 4,2 and a 6,2 fluorotelomere side chain, based on the fragments that we see in the MSMS spectra. These mixed chain dipaps are often present as residual byproducts from the manufacturing process to produce these dipap chemicals. Next, I'm going to talk about PFAS and product packaging. So, because of the great ability of these compounds to confer oil, stain, and water repellency in food contact material, PFAS have been widely reported in these products in Canada, the States, and Europe. And in a European survey that looked at food contact material, every participating country had samples with PFAS, with some at very high total organofluorine levels, up to 60 times higher than the indicator threshold set by the Danish authorities as a marker for intentional PFAS addition. However, the news isn't all bad though, because in Europe, the regulation has motivated actually a lot of companies to shift from PFAS to non-fluorinated alternatives. So, as an example, in Denmark, a PFAS application in food contact materials has been banned since 2020. And in this survey, none of the French fry bags from a popular fast food chain restaurant in Denmark had showed any intentional PFAS treatment, which was in contrast to samples from the same restaurant chain in the Czech Republic, Germany, and the UK. So, and we're also starting to see a lot of similar regulatory actions taking into effect in North America. As of today, 11 US states have adopted bans on PFAS in food packaging, some of which have already started to take into effect this year, with more to come in 2024. And another positive sign that we're seeing over here is that there is an increased emphasis on supply chain transparency, where states like Washington and Maine are now mandating that companies must disclose PFAS as an ingredient in their food packaging products. So, in response to these regulatory trends, we have developed targeted methods using both the X500 QTOF system and the 7500 system to monitor for the presence of PFAS known to be present in food contact materials. In China, for example, PFOA and PFOS levels in food contact material are regulated according to their national food safety standard. So, our colleagues in China have developed an MRM-HR method to quantify 17 different PFAS and also confirm their presence by library matching using the X500 system. On the right here, we reported the presence of a suite of DIPAPs on the 7500 system, including this mixed chain length one, like what I discussed earlier in the previous slide, in almost all of the food packaging materials that were analyzed. And this was not surprising because considering these chemicals have been previously reported in numerous food packaging monitoring studies, as well as in paper sludge and paperboard materials. And finally, I'm going to talk about PFAS in food. So there's been a number of food surveys that have recently highlighted food as a very important contributor to dietary exposure to PFAS in humans. The upper left figure here shows the distribution of PFCAs and PFSAs observed in various seafood samples in a survey conducted by the US FDA. I know it's very hard, it's a bit hard to see here, but the y-axis on this graph for the clam profile, this is actually 10 times higher than all these other samples, which indicates the clam had the highest total PFAS concentrations as compared to all the other seafood types, with PFOA as the dominant congener observed at levels as high as 20 micrograms per kilogram. And these results actually led to the distributors of these canned clam samples issuing a voluntary recall for these products. This seafood survey actually stemmed from an earlier study also conducted by the FDA, which is called the Total Diet Study. And this study was testing foods from the general food supply. And in this study, they actually found relatively low levels of PFAS in a small number of seafood samples, as shown here in Table 2. But what's really interesting is that this disparity that we're observing between retail seafood that are purchased in grocery stores compared to locally caught seafood was made even more pronounced in a paper that was published earlier this year from Duke University, which analyzed data over 500 composite fish samples collected by the EPA. And in this study, they showed that the median levels of freshwater fish were actually greater than 250 times higher than levels in the commercial retail fish samples that were tested by the FDA Total Diet Study. So this really indicates that there's a pressing priority to establish a harmonized fish consumption advisory across the state. And as I'm not sure if everyone has had a chance to see the news report, yes, from last night, but just hot off the press, actually from 2 days ago, there has been a health alert issued after PFAS was found in 13 state parks in Massachusetts, which resulted in the local authorities to issue new fish consumption advisories set in those locations. So here at SciEx, we have also started to internally develop methods and also work with our collaborators to develop both quantitation and screening methods for PFAS analysis in food. On the left here is the concentration profile of different PFAS observed in various food samples. And no surprises here, we also see higher levels of PFAS in salmon and tuna as compared to the other samples, as well as in beef. And I think the detection in beef here is actually really interesting and relevant to last year's report of PFAS found in cattle raised on farmlands that have been using biosolids heavily contaminated with PFAS. And this practice was used as a fertilizer to amend their soils. And on the right here, this is showing data from analysis of a meat sample that was provided to us, and it was sourced from livestock raised on on similarly contaminated farmlands as well too. And using SWOT DIA, or data independent acquisition, on the Xenotop 7600 system, we were able to confirm the presence of various PFAS, for example, such as this PFAS shown here, using this non-targeted screening workflow. So for this year, we have planned a lineup of very exciting projects with our collaborators to explore even more important exposure areas in this PFAS life cycle, such as wastewater treatment plants, compost and landfills, more consumer products, air and house dust, and as food as well too. And we're hoping to present results from some of these studies in the second part of the summit later this year. I'm also excited to announce that a group of us have also joined the AOAC International Working Group for PFAS to work with really an interdisciplinary panel comprised of government, industry, and academic representatives on developing standardized and analytical methods for PFAS testing in food. So please stay tuned for more on this topic. So lastly, if you haven't seen already, SciEx launched a website dedicated to PFAS last fall. And it's an evolving website containing a huge amount of information that would hopefully help guide you on your journey to learn more about PFAS and also access our technical content on how to analyze these chemicals. So two things that I do want to draw your attention to is our repository of PFAS technical notes, some of which I've highlighted in one of my earlier slides. And this section allows you to filter based on the matrix type and instrument type, making it very user-friendly and easy for you to access the content that is most relevant to your needs. And secondly, on the website, we also have profiles introducing some of our internal PFAS experts, which highlight both our past and our current experiences with PFAS. Which actually brings me to my final slide here. So what I want to say about this is that one thing that I think is unique to SciEx with respect to PFAS is that this team of internal experts who all have very extensive experience both in LC-MS analysis and PFAS, what really differentiates is that most of us did not only just start to work with PFAS since joining SciEx, but a lot of our experiences actually trace back to our graduate and postgraduate days in academia, stemming from as far back as at least 10 years back when most of our PhD thesis or postgraduate work were collectively about PFAS, as you can see from the number of publications that we have in each of that are tied to each of us. So in addition to having the technical background on the analysis of these chemicals, we are also well versed in the scientific background on PFAS, with respect to their history, chemistry and toxicology, and their fate and transport in the environment as well too. So with that, I'd be happy to answer any questions you might have.